Hi everyone, good morning. My name is Teun. I'm a scientist with Louis Croyer at the Biohub here in San Francisco. And today I will be sharing our recent advances regarding cell tracking, both from the algorithmic point of view and visualization. Because in our group we are trying to understand development, and ideally at the single cell level. And in the video here you see a developing zebrafish embryo during its roughly first day of development, imaged in one of our light sheet microscopes. And basically development, and you could say life in general, is all governed by dynamics. And in order to understand and study these dynamics, we want to follow the individual cells in this, during this process. And to follow these cells, we use cell tracking algorithms. And cell tracking, as the name implies, is basically just following single cells in a microscopy video. And we humans, by eye, are extremely good at this. But for computers, it's quite challenging still. So that's why we're working on this. And in today's presentation, I will go over two methods we worked on. One is ULTRAC, and the other one is INTRACTIVE. ULTRAC is the method, the actual tracking algorithm, so the algorithm that does the tracking itself, developed by Jordao in our group. And INTRACTIVE is the way to visualize your cell tracking data. So let's start with ULTRAC. Basically, there are many cell tracking algorithms out there, and they have been for a long time, but it's still very challenging. And tracking is hard, mainly because segmentation is hard. Because tracking is sort of done traditionally in a two-step process. You first have to find your cells, and once you have found them, you're going to basically link them across time. And especially this first step is challenging, especially in like a developing zebrafish embryo, where it's very dense and cells are hard to distinguish from each other. The problem of this two-step approach is that if you make a mistake in the first step, the finding of the cells, like for example in the middle there you see like 4 cells sort of grouped together, that means that in the second step, the linking between frames, you will definitely make mistakes because it's basically seen as one cell. And so basically to solve this, what ULTRAC uses is a joint approach. So instead of doing first segmentation and then tracking, we do it sort of simultaneously. And the idea is that basically when you have an image of a microscopy image, in for example of a nuclear label, and you see this blob, you could say this is just a single cell. But it could also have been just 2 cells very close to each other. So instead of making a decision before the tracking, we do it simultaneously with the tracking. So what ULTRAC does is it uses hypotheses. It could have been 1 cell, outlined in white, could also have been 2 with the gray line. And instead of making a decision, it keeps both hypotheses alive. And to make a decision, it basically does the tracking. It looks across time. So in this case, when, for example, in the previous time frame, you clearly see 2 cells, and in the time frame after also 2, it was very likely that also in your frame of interest, it was also 2 cells. So by doing the tracking at the same time as the segmentation, basically you improve both steps. That's the idea of ULTRAC. And apart from that, it's very optimized to work on large data sets. So terabytes of imaging data can just be tracked in a few hours. It works on 3D data, multicolor, etc. And it can handle this segmentation uncertainty. Basically meaning when you don't really know what the cell is or what not, by temporal consistency we can solve this by doing the tracking at the same time. And that's ULTRAC developed by Jordao. And with this tool, we can now track all these cells in our developing zebrafish embryo. So here you see a part of the embryo, and we zoom in on a single cell, and in the red line is basically the trajectory of that single cell. And we can do that for a single cell, but ULTRAC does it at the same time for all cells in the zebrafish embryo. So what you see come up here is like all these colored trajectories are all the paths of every cell in this embryo. So we can do this now simultaneously for all 10,000 plus cells in the embryo, and basically fully map out how they all go their way during development. So where do they go, where do they come from, when do they divide, etc. So this gives us a very rich data set to study these dynamics in these first crucial hours of development. So that's the tracking part, ULTRAC. So now you did your tracking, you have your trajectories, you want to visualize them. And to do that, we developed INTRACTIVE. Because the problem is these data sets are huge. It's basically 3D trajectories in time, so it's sort of 4D data. You have millions of cells over all your time points. So you first of all need to download your data. You need software to even open it, for example, Napari, but you need to basically, it's not very interactive, so there are quite some limitations. So to make this very user-friendly and interactive, we worked on a tool we call INTRACTIVE. And INTRACTIVE is an interactive tracking visualization tool that lives in the browser. So it basically looks like this. So it's just an application living in your browser. You'll see you have a big canvas that displays your cell tracking results. There's a time slider in the bottom to go basically across time. And on the left side, there's a sidebar with buttons and toggles to customize your visualization. And with INTRACTIVE, it's basically designed to look at large 3D cell tracking data sets, or any tracking data set. It fully lives in the browser, so no need to download the data or download any application. And with it, you can do interactive lineage tracing. So basically, in this case, again, a zebrafish embryo, by moving the slider, you go through time, you can pan, rotate, zoom, study the entire point cloud. And you can even select some cells, and when you then go back in time, you can exactly see where those cells came from. So you can select some cells in your tissue of interest and see where did they come from, etc. So another example is, for example, the Drosophila embryo. So this is another lychee data set of a developing Drosophila embryo, and when you go through time, you can see all these cells massively moving during the gastrulation. In interactive, you have selection tools. So for example, you can select some cells at the top of the embryo, and by going back in time, you see, okay, those cells, where did they come from, and where did their parents come from, etc. And interactive is developed with the idea of being very collaborative and open. So for example, if you are studying this tracking data set, and you have a view and you're like, I want to share that with a collaborator, for example, what you can do is you can basically go to the point you want to share, you press a button that copies the URL or the link to that viewpoint. This URL you can send to your collaborator or a colleague. They open it on their computer, and they go to exactly the same viewpoint that you copied the link from. And there they can basically see the same thing as you saw and continue their investigation from there. And to pull this even further, we now incorporate these links in our papers. So for example, LTrack was recently published, and there are many figures are around cell tracking, but it's just a 2D capture of a large 3D cell tracking data set. You don't really get it. So what we do is in a caption, we can now embed a link. When you click on it, it basically brings you directly to the data, and you can play with the data sets yourself that were part of this figure. So this is ideal for readers, but also for reviewers, for example, to get a better idea of the data they're looking at, apart from just screenshots. And since it works in the browser, it works in any browser. It could be your laptop, but also a tablet, iPhone, iPad, etc. So you can just have it basically anywhere in your pocket and look at this. Apart from just basically the zebrafish that we image, we asked our collaborators for the best data sets out there of embryogenesis imaged with light sheet. And with this, we built the virtual embryo zoo, which you can visit via embryozoo.org. It's basically a showcase of currently like 6 model organisms imaged during their first stages of development. So we have not only zebrafish, but also mouse, C. elegans, Terbolium, etc. On this website, you can basically inspect those data sets, and with a single click, you open them basically in interactive, and you can visualize them. So in this case, you can again look at the zebrafish. And this is a tool that is very nice for example, us to share our data, also for educational purposes, to give students the opportunity to delve into embryogenesis at a single cell level, but also see differences between different organisms. Personally, I like to show it to my parents and friends, like, look, I can finally show you what I'm doing, because it's just on their phone, and it's very hands-on, so it's very useful during Christmas, etc. Recently, we added extra functionalities to interactive. You can now color cells based on certain features to make it more information-rich. For example, in this case, we can color the cells on, let's say, the direction they're moving. So different colors are different directions, and you can see different tissue flows. We can basically color divisions here in red, and you can see these nice methodic waves during development. Or we can color, for example, cell density or local structure. There are many features. You could be a gene of interest that you labeled. Anything that's in the data that you can calculate from the data, you can overlay as a color to make the visualization even more rich. So up until now, these are all data sets from the virtual Embryo Zoo website. And they're out there, you can play with it and go crazy. But we also wanted to make the tool as modular as possible to look at your own cell tracking data. So let's say you do your own cell tracking data, it could be 2D or any modality, and you have it on your computer and you just want to visualize it. So instead of using like matplotlib, 2D plot, very static, you can just basically open it in interactive to play with it yourself. So we have a list of options how you can do this. One would be the command line via the terminal, but we also have more user-friendly like UI-based options, for example, from the Napari or from a Jupyter notebook. So I have a very quick demo just to show you how that works. So let's say from Napari, let's say you have a Napari viewer and you have your tracking data in Napari in the tracks layer. What you can do, you can open up the widget, the interactive widget, and with a single click of a button, the data from Napari is ported to your browser, and you can get the full interactive functionalization with all the coloring, etc. in your browser. Second thing is from a Jupyter notebook. Let's say you work in Python, you have your data in your notebook. With a single function call, you do the same thing. It opens a browser, it pops up a window, and you can inspect your data dynamically and interactively on your own computer. So it is not hosted anywhere, it's all living locally just to play with. So, and lately we added a fourth option. You can now do it also from Fiji, like in TrackMate. So when you use TrackMate in Fiji to do tracking, in the end of the tracking there's a button, visualize in interactive, and you can even more dynamically visualize it. So those are the two tools. We have Ultrack to do the actual tracking, and interactive to visualize your cell tracking results in a very interactive manner. And we're happy to share that both methods recently got published in Nature Methods in 2 papers, I think last week, so it's very recent, so please go ahead and check them out. All the code and tools are fully open source on GitHub, so check it out. Ask us if you have any questions or get started with it or find any bugs, we're very welcome to help you there. And of course the Embryo Zoo website to play with these embryogenesis data sets. That leaves me with acknowledging everyone in our group, in the Royal group, also investigators, like collaborators, both within the Biohub and at CZI, and thank you for your attention.
Beautiful, thank you. All right, any questions? Let's see over here.
Hi, yeah, thanks for the talk. I thought that was really cool and impressive, all the things you're doing. Yeah, so my question is, how well or how precisely is this tracking algorithm? Like, could it track to the nearest 100th of a pixel, for example?
Can you repeat the last part? Can it track?
Could it track like to 100th of a pixel precision, like sub-pixel tracking?
Right now, it is instance tracking, so you need to have, you start with a segmentation of what you define an instance. Could be, in this case, cells, could also be a molecule on the membrane, anything that you define as an object, and that can be tracked. So, of course, it's way better than any other tracking algorithm. It's still not perfect, of course, because you're still limited by, biological factors. But it's basically just like, right now, based on many metrics, the best one out there, especially for these huge ones where you also want to do the tracking in a reasonable amount of time.
Okay, yeah, that answers my question. Thanks.
Yeah, I was just wondering if you could give a basic idea of like how long the tracking algorithm might take to run for these very large data sets. I guess it depends on what resources you have, but
yeah. So the one I'm showing many times, like the zebrafish, that's like a 24-hour imaging session. Data set is around 10 terabytes. I think it's 1,000 time frames. That takes, but when you have like a nice GPU cluster, a CPU cluster on your, then it takes, this case took like 5 hours. We also tried on a laptop, like just a normal laptop. It runs the same thing, it just scales to whatever hardware you have, then it takes a week. So it depends on how much you have. But even in a week, for this amount of data, I would say it's not bad. But as long as you can parallelize it, it's already way better. Yeah.
So in the case of the self-hosted interactive sessions,
yes.
What does it take to host them openly for others? So if I wanted to have a mirror with data for some other lab, let's say.
Yeah. So the application itself, when you host it yourself, you host it locally and it's fully yours. The application that you go to when you click on any link that I showed you is basically a statically hosted one on GitHub. The only thing you need is that if you want to have it publicly, your data must be somewhere. So it must be on a repository somewhere where anyone can access it. Could be on, in our case, we have our website where the data is published. Could be GitHub, Google Drive, where it's open. As long as anyone can access it, it should be clear. Yeah. Yes, good question.
So one of the issues that we face when playing with some of these things is you change a little bit of imaging conditions and it's like a whole new training process for the segmentation, for the tracking. So, you know, I don't know how this algorithm does in terms of that, but like, you know, how does it do in terms of that, and what are the limiting? So can we get to the point where you just give it something and tracks it without having to go through 3 months of optimization?
Yeah, ideally yes. So currently there are of course still parameters involved. So you start with a segmentation or a list of segmentations. There are still like 3-4 parameters you need to sort of tune to get it right for your data set. Ideally, like this is for example fully deep learning free. You can use deep learning to do segmentation, but it's not necessary. It can be fully image processing. Once we go to a more deep learning framework, then you can basically have like data sets where you train these parameters on and you can find the best ones. Or right now we also have basically options where you basically annotate a small section of your data set, and once that is basically have like a sort of little ground truth data set, by basically doing like a parameter sweep on that, we can find like the parameter ranges that helps you guiding it. But yeah, there's still some, as any method, some tuning involved. But ideally that becomes less and less over time. Yeah. Thank you.
Yeah.
Thank you.